So here I'm going to go through some of the anatomical and molecular records related to evolution. Think of it as some of the macroscopic and some of the microscopic records related to evolution. First off, we have the presence of vestigial organs. Structures that are no longer in use, such as the human appendix and wisdom teeth. Apes have a much larger appendix that's involved with the digestive process. We have these structures and we can easily live without them. This is evidence that they may have been used at one time, and now if we're born without them, we have no negative effects. Molecular records, well, evolutionary distance is one, for example. New alleles arise by mutations that come from predominance through favorable selection. So in this case, we have this kind of population before. After selection, we have the favor of the red circles instead of the yellow. This could relate to the resistance level of something going on in the environment. We also have the example here. For whatever reason, the dark gray is favored over the white coloration, so we have this change over time. Now we look at specifically these molecular records and this kind of divergence that is occurring. We can look at kind of degrees of divergence or how dissimilar two species may be. Evolutionary changes involve the continual accumulation of genetic changes. Distance-related organisms accumulate in greater number of evolutionary differences than closely related ones. This divergence is seen among vertebrates in the 146 amino acid hemoglobin beta chain. So what does that kind of mean? Well, looking at hemoglobin, which humans, monkeys, dogs, birds, frogs, and even sea lampreys have, seeing the amount of amino acids that are different between this hemoglobin polypeptide compared to the human. So in this case, the monkey has 8 difference, the dog has 32, the bird is 45, the frog 67 different amino acids, and then the lamprey 125. This indicates that while all of these organisms have hemoglobin, the lamprey is by far the most dissimilar because of its high degree of variance. And the frog and the bird are more distantly related to humans than the dog or the monkey. And we're looking at our molecular clock and the difference between these. This same pattern of divergence is seen with DNA sequences, such as that of the cytochrome C gene. So what does this kind of refer to and mean? Well, here we have millions of years and nucleotide substitutions. Well, changes appear to accumulate at a consistent rate. This phenomenon is referred to as the molecular clock. This means as mutations are occurring at a relatively consistent rate. Note different proteins evolve at different rates. So the more complex we have, the least amount of differences compared to the greater amount of differences we have here. So here we have, again, the human, the pig, the duck, the snake, fish, and moss species in yeast. We have this large divergence here. So looking at the molecular clock, the degree of nucleotide substitutions that are occurring over this period of time, we're seeing some sort of consistency to that pattern over a long period of time, indicating this molecular clock. We've also something called homology. So the eyes of these organisms are not homologous. That's the first kind of key point. But the genes controlling the eyes are homologous. So let's kind of look at this in more detail. When we are comparing the genetic similarity to protein similarity of an eye control gene across species, that's what we're looking at in specifics here. The mouse is the comparison standard. So for example, the genetic similarity and the protein similarity. So protein similarity to the mouse between the eye of the mouse, the shark, the squid, and the flatworm is all 100%. However, what's different is the genetic similarity to the mouse. So this gets into genotype and phenotype. So genetic similarity to the mouse, the shark only has 85%, the squid a little over 78%, flatworm 71.6%. So we're seeing that there is some genetic variability, even though this protein is consistent throughout. So this indicates, remember, there's multiple ways you can make the same protein, get to the same amino acid with different genetic code, different RNA, essentially. Within this molecular record, looking at 500 million years ago, an ancestor involved a switch for eye development. So probably a simple type of eye, one pigment cell, one photoreceptor. This gene was passed on to his descendants, and these descendants involved more complex eyes. Well, if we have just kind of light and dark sensing, well, that's one way. But if there's more complexities being developed, this is a way that that gene can be modified to development of kind of the complexity to the eyes, for example, that we have. We can sense a lot more than just light and dark. We have lenses, colors. There's a lot of complexity just within the eye. This again shows that kind of basic, there's nerve fibers and photoreceptors. We'll get into development of a retina, getting development of cornea, development of lens, being able to kind of sense objects at different distances, being with night vision, depending on what animal you're looking at, a lot of complexities here. Looking at dinosaurs as well, we could see this high degree of complexity that is occurring in divergence between them all. We have definitely a change that occurred where a lot of them went extinct. So why did that happen? What they clearly did not best fit that environment, but this just shows you that progression over time. Now, molecular evidence, we have DNA hybridization, it's called, where DNA is separated with sufficient heat to create single strands. These then closely related sequences will join together, or what we call hybridize. They'll stick together more strongly since there are more complementary bases. So we kind of take the DNA, we kind of separate it out, and we see what it may stick to. The closer the sequences are related, the higher the melting temperature, the TM, will be required to separate those. This is just a kind of a way to tell this hybridization how similar the genetics may be, because if they're more similar, they're going to be bound tighter and take more heat to get them to separate out. Now, whale evolution is something else to kind of look at. The evolution of whales, beginning with their ancestor, their descendants of terrestrial animals return to the sea. So this is another example of evolution doesn't mean getting more complex necessarily. Whales' distant relatives may have been land-based with walking, and they may have slowly reduced those limbs to become more flipper-like and to become more fin-like to be able to best fit the aquatic environment versus the terrestrial one. There's a lot of evolutionary critics. The 7 principles of objectives that they have here, it's not solidly demonstrated, there's no fossil intermediates. There's intelligent design. Evolution violates the second law of thermodynamics. Proteins are too improbable to form. All these objections are really without a great scientific merit when you start looking at them to be kind of supported. A lot of people bring up religion related to evolution. This is just looking at Pope John Paul II in November 19th, 1996, an address that he made regarding evolution, because the Catholic Church and evolution tend to be thought to be very much conflict. So he stated that today, almost half a century after the publication of the Euclidean, new knowledge has been led to the recognition of more than a hypothesis in a theory of evolution. It's indeed remarkable that the theory has been progressively accepted by researchers following a series of discoveries in various fields of knowledge. He goes on to say the convergence, neither sought nor fabricated, of the results of work was conducted independently and is itself a significant argument in favor of the theory. So keep in mind it is a theory, but in science, theory is a very well-established, well-supported idea or concept versus a theory that we may be familiar with more in common everyday world.